Hello, welcome back to another another science stream, and today the main topic is or are viruses. Hopefully my mic works. Yes, it does. So, let's start with what is a virus, okay? So, a virus is a thing or a microscopic organism that infects hosts and like animals, humans, plants, and etc. And they can't reproduce without a host, so that's the main difference, I think, if you compare it with like stuff like bacteria and all of that. And the main two stars of the show for today would be VSV and adenovirus. Okay, so let's start with the difference in their expression strategies. So wait, I'll write it in a different way. One moment. Here, nope. Bigger eraser. Thank you. Okay, there we go. So adenovirus is adenovirus. It's a naked DNA virus, okay? Adenovirus, it's a naked DNA virus, meaning it does not have a lipid envelope and it carries a DNA genome. Again, adenovirus is a naked DNA virus, meaning it does not have a lipid envelope. Meanwhile, VSV is enveloped. It's enveloped with an RNA genome. So again, if I just make a table between adenovirus and VSV, adenovirus would be naked DNA genome, naked virus. 1 second. And this genome is DNA genome. Meanwhile, VSV would be it's enveloped and it uses an RNA genome. So that in itself is already a very big difference if it has DNA or RNA. And you have some key discoveries from virus research. So for example, studies on small viruses like SV40 or papillomavirus and adenovirus, those led to different discoveries like mRNA splicing, mRNA polyadenylation, and transcription control mechanisms. And that's how the viral research has significantly increased our understanding of the gene regulation in eukaryotic cells. So some questions that I would like to remember the answer to would be these. Let's see. Wait 1 second. This part. Adenovirus versus VSV. Adenovirus is a naked virus, meanwhile and it uses a DNA genome. Meanwhile, VSV is enveloped and RNA genome. The questions I mentioned earlier would be, how do adenovirus and VSV initiate gene expression? How is viral expression studied in the lab? What is a viral vector? And how are recombinant viruses made? And what is genetic complementation? And why is it useful in viral research? So those are some questions that I would like to answer. Alright, so just a bit of the history. Viruses cause many serious human and animal illnesses. So for example, you have smallpox and some virus associated diseases, but thanks to the vaccine, smallpox was eradicated at around 1980. But because of its severity and it's very contagious, the virus remains a bioterrorism threat. So, and another example is pox viruses like monkeypox. Those could emerge and pose public health risks, which could highlight the ongoing importance of virus surveillance, actually, and an impact of vaccination on viral diseases. They just really lessened the numerous viral diseases, so get your vaccine. And some recombinant virus technology in health. The vaccines deliver antigens safely, and in gene therapy, they serve as vectors to introduce therapeutic genes. And as oncolytic agents, they are designed to selectively infect and destroy cancer cells. Meanwhile, in research, the recombinant viruses are used as like tools to study gene function, replication, and immune responses. Right, so pandemic preparedness and zoonotic viruses. So many viruses that cause outbreaks in humans originate in animals, particularly the zoonotic RNA viruses. So if you study these animal viruses, those that could help us be more prepared for different pandemics. And by using the modified viruses, scientists can develop vaccines and like therapies and tools to study viral behavior. And SARS-CoV-2, the virus behind the SARS-CoV-2, aka the virus behind the big COVID-19, is a prime example of a bat origin virus that made the jump to humans. So this is a bat. That's Batman. Yup, that's the bat. And this is a human. Nice. Okay. Next is 2014 to 2016. Around that time, Ebola happened around in West Africa. 2014, Ebola, and that was the largest record on record because it yielded around 28,000 cases and 11,000 deaths. And then there is also the changing patterns in filovirus outbreaks, aka the Ebola and Marburg. It's becoming more frequent. And then if you compare adenovirus and VSV here, if you compare adenovirus and VSV, they differ in structure and genome type. I mentioned before, adenovirus naked and DNA, meanwhile VSV would be enveloped and RNA. So, but despite the differences between them, both viruses can be used as like vehicles for vaccines because they have the ability to express foreign genes. However, viruses must infect a living host to replicate. So that's a very crucial detail because they are intracellular parasites. Okay? They are intracellular parasites, meaning they infect living cells to replicate, and that requires breaching the host defenses, aka entering permissive cells, and you would need to uncoat the genome and hijack the machinery to replicate it and assemble the viral components. And after replication, the viruses would exit the host to infect new cells, so which would often damage or kill the host cell in the process. So just as an overview for the viral cycle, erase. As an overview for the viral cycle, you have number 1, entry. Number 2, uncoat. COAT. Number 3, genome replication. Number 4, mRNA and protein synthesis. Number 5, assemble the new virions. Assemble the new virions. And then 6 is release. So again, entry, uncode, replicate the genome, mRNA and protein synthesis, assemble the new virions, and release. Let me write that again, because these are the steps. So number 1, entry, and then second, they need to uncode it. Third would be replicate the genome, like Taju, Kagi Bunshin Jutsu. Fourth would be mRNA and protein synthesis. 5 is assemble the new virions. The new virions. Number 6 is, what's this one? Just exit, right? Yeah, release. The 6th is release. And for the exact steps depend on whether the virus has DNA or RNA inside, aka if it's adeno or VSV. So for example, VSV replicates the cytoplasm without entering the nucleus because it's RNA. Meanwhile, DNA viruses like AD requires nuclear access for replication. So the table is increasing. Again, let's write the difference. Adenovirus versus VSV. Adenovirus would be not enveloped. It is using a DNA genome, and then this one is, and then how does it, and does it need access? Yes, it require nuclear access. Meanwhile, VSV is enveloped. It's RNA and replicates the cytoplasm. The cytoplasm without entering nucleus. And how do you let's see, how do you study virus infection in the lab? Well, viruses must replicate in living cells, so that's why you use cell lines like Vero or 293 cells. The lines are transformed and just grow indefinitely, and thus they support the replication. Okay? And cultured cells actually mimic the tissue of origin, and that's why they're used to study virus entry and replication and other effects. Cells can be diploid, like wherein they would have a finite lifespan, and they can also be continuous or immortal. There's a thing called CPE, aka cytopathic effect by virus infection, and CPE would be referred to the visible changes in the host cells caused by virus infection, and those changes could vary by virus and cell line. So yeah, it depends. So for example, VSV causes the Vero cells to jump, to round up and detach, while the measles virus forms a syncytia. So CPE is a useful readout for infection. Okay, so again, cytopathic effect, what is it? It's the visible changes in the host cells caused by the viral infection. Again, what is the cytopathic effect? It is the visible changes on a host cell caused by the actual virus infection. Okay, next, virus growth in cultured cells. This one would be kinetics, okay, because it could be 1 step, it could be 2 step. So you have virus growth in cultured cells. And 2 types of infection condition. You could be, let's see, you could be 1 step, okay? I'll do red for the 1 step. First, you have attachment and entry. Wait. X-axis. X-axis would be hours. Y-axis is the viruses in the medium. Free viruses. Viruses at medium. So first, attachment and entry. So the amount of viruses in the medium would lessen. Eclipse happens, and then maturation, and then plateau. So again, attachment and entry, eclipse, and then you have the maturation. Alright, now this is 1 step, but if it is 2 steps, you have similar starting point, attachment and entry, eclipse, a bit of maturation, another eclipse, and maturation again, and then plateau. So you have maturation, plateau, and then maturation again. So that is a 2-step curve. So the difference in 1 step or 2 step is, in the 1-step curve, all of it is infected, like all the cells are infected at once. Meanwhile, in the 2-step curve, in the multi-step, a low virus dose allows for consequential, sequential rounds of replication. So again, 1 step versus 2 step. 1 step would be all the cells are infected all at once. Meanwhile, the 2 step would be you have multiple rounds of maturation. Nice. Nice. Next is the plaque assay, aka measuring the infection virus. So for this one, you have it quantifies how much infection virus is present, and it involves serially diluting serially diluting a virus stock and infecting a cell monolayer. Alright, let's write it here. Again, a plaque assay measures the infection virus. The way it works is you dilute a virus stock, and then after that one, you dilute first. That would be lots of test tubes, like dilute, and then dilute, and then you put these diluted ones into like a very visible plate. The cells would be overlaid on a semi-solid medium to limit the spread, and you can see a very clear area. So counting the plaques gives the plaque forming units per ml. So that's how a plaque assay works. And yeah, you dilute and then you count. That's a plaque assay. And you have multiple uses for the plaque assay, because they're used to analyze the phenotypes and the temperature sensitivity. So for example, the VSB mutants would form smaller plaques and at higher temperatures. So again, smaller plaques, smaller size at high temp. And the plaque morphology would help characterize viral fitness, and the function of the mutated genes. So for its structure, sticky note. Virus sticky note. For its structure, here we go. Viruses are made of a nucleic acid genome enclosed in a protein shell called a capsid. Alright? It's enclosed in a protein shell called a capsid. And let me just draw that really quickly. You have this one. This is a protein shell, the capsid. Wait, let me point an arrow. Hello, Yaji. Yes. Early bird, something the worm. No problem. Yeah, these are the viruses. Right now we're talking about the naked viruses, aka naked viruses. Ohayo gozaimasu. Welcome to the stream, by the way. I think this is your first time here? Adenovirus. Oh, I have to introduce myself. Wait. Hi, I'm Millennium, Isekai'd, Mask Knight, Empress Scientist from Earth so far. And yeah, I do Pokemon, Pal World, and science. And today we are learning about viruses, aka adenovirus and VSV. So adenovirus would be, they're the naked viruses. It uses DNA as a genome, and then it's, what's this called? The third detail is it might need some nuclear stuff, entry help to come in. Meanwhile, VSV, I'll draw the insides later. Meanwhile, VSV would be, it is enveloped, okay? Enveloped. And then it has the RNA as a genome, RNA. If you draw it right now, viruses are made of a nucleic acid genome enclosed in a shell. This is the capsid, aka the protein shell. And then you have the nucleic acid over here. You have the nucleic acid here. That's the DNA. Alright? Meanwhile, your dear VSV, you have the RNA genome inside, but you have a lipid envelope outside. So that's the difference between the VSV and the adenovirus. It has an envelope outside, and many types of viruses infect animals. So because animal viruses vary widely. Adenovirus and rota, again, they're naked, meaning they lack the lipid envelope. Meanwhile, herpes and influenza are enveloped. Okay, so as example here, I can write it. Rota and adeno are no envelope. VSV, the examples would be herpes and influenza. That would affect their stability and transmission. Alright? So next, gene expression depends on the genome type. Because viruses must generate mRNA to produce proteins. And depending on the genome, the strategy would depend on the genome. Alright, and DNA viruses like adeno, so remember, adeno here, they can host the RNA polymerase. Okay? Meanwhile, RNA viruses should carry their own RNA polymerase. So that's why they do it outside. That's another main difference that should not be forgotten. Okay, let's remake the table. Let's make the table again. Erase these. Here. Erase. Hi. Okay. Adenovirus, and here, and then VSV. So what's this one? This is a naked virus, no lipid layer. This one uses DNA, and then this one goes inside the nucleus, and then it can uses the machinery. DNA viruses like adenovirus can use the uses host RNA machinery. Meanwhile, VSV has a lipid layer, uses RNA, it stays outside, and it needs its own polymerase. Okay, don't forget that one. I will also picture the table. Wait. There you go. Photo taken. Okay, now that's finished. You have something called the Baltimore classification. They group viruses into 7 classes by genome type and the mRNA generation strategy. Alright, here. The group, where's the 7 type? Now, there you go. DNA, I'll write it first in order. DNA genomes, and then RNA genomes. RNA genomes. Okay, perfect. You have here dsDNA, ssDNA, and then ds/ss gapped DNA. The DS I remember is Nintendo DS. And then RNA would be, okay, dsRNA, ssRNA, SS positive. Then you have ssRNA negative, and then you have ssRNA positive with reverse transcriptase. A lot. That's a lot. Anyway, let's write it again. You have DNA genome viruses, DNA genome, and RNA genome. You have dsDNA, ssDNA, and then ds/ss gapped DNA. RNA virus would be dsRNA, ssRNA positive, ssRNA negative, and then ssRNA positive with reverse transcriptase. Slay! Nice! So the distinctions determine the replication site and enzymes needed. Beautiful. And we talked about the different gene expression. Now, expression from a viral DNA genome. Then the adenovirus they rely on transcription control mechanisms to regulate gene expression. And the expression pattern usually follows a cascade, aka like immediate early, and late genes. So, and gene activation is usually in a very controlled manner. Then you have temporal control, aka the timing, the life cycle of it. And you also the different temporal control also ensures that the host cell is not overwhelmed prematurely and that the viral assembly occurs efficiently. Alright. And the gene expression is categorized into, like I mentioned before, immediate, early, intermediate, and late stages, and usually it's RNA polymerase 2 that does the transcriptional control. Hi. Okay, different genes. Ta-da-dum. You have the E1B, E2, E3. You have late transcription. It requires viral DNA replication. And then you also have some different drug studies And how can you make, what's this called? How can you make the gene delivery vector? It's a good question. Okay, so it has a rigid structure. You can't stuff a longer genome inside, and you can't just add another gene. Okay, don't just add another gene just for the sake of adding another gene. So one strategy for gene delivery would be to replace or just change some of the regions of it. So for example, you earlier mentioned like it has E1A, E1B, and you can just remove one of those or something at some point to make space for the other ones. Depends on what you want to do. And then you also have recombinant viruses. Okay, a recombinant virus can be recombinant adenovirus. You can create it by how? Let me see. You can create a recombinant adenovirus by cloning its genome into a plasmid and replacing E1 with a gene of interest. What does that mean? Okay, one moment. So you have this gene. This is the adenovirus genomic clone. And then we clone it but add a foreign gene over here. Foreign gene. And then you put it into a plate, or you transfect it into like your cells, and you watch the cytopathic effects emerge. Again, what is a cytopathic effect? That would, what's this called? Those are the effects on a host because of the viral infection. Okay, and then you can just use different vectors for that. Now, moving on to VSV. It's enveloped. It has a negative sense RNA genome with 5 genes. NPO. What's the 5 genes of it? Here. It's enveloped, and it needs to use its own system. It has the NPMGL genes, NPMGL. Yup, those genes transcribed sequentially by the RNA-dependent RNA polymerase. And compared to the adenovirus, the DNA viruses, these VSV RNA viruses are harder to manipulate because it is RNA. Negative sense RNA should be replicated or transcribed into mRNA by a viral polymerase. And to insert a new gene, the scientists should integrate it into the RNA genome while maintaining the transcriptional gradient and ensuring correct replication and encapsulation. Okay? And yes, negative sense RNA is complementary to mRNA, so it cannot be translated directly. It lacks a 5' cap, and it's not recognized by the host RNA polymerase or ribosomes. All right. Okay, now the next question would be like, what does it mean, the positive sense or negative sense viruses? Good question. So you have RNA viruses. Nope. I'll write it at the bottom. You have RNA viruses here. You have the dsRNA. Dude, I cannot write. One moment. You have the dsRNA, ssRNA positive, ssRNA negative, and then ssRNA positive with reverse transcriptase. So, positive sense can just use their RNA directly as mRNA. So no issues there. they can just use their RNA directly as mRNA to translate that into a protein and then cleave into other proteins. So they bypass the need to transcribe their genome into another form. Meanwhile, negative sense viruses like VSV, measles, mumps, and Ebola, they need to their negative sense RNA cannot be immediately translated because that's not recognized by ribosomes. So first, they have to be transcribed into complementary RNA, complementary mRNA by their viral polymerase, because they have an absence of a 5' cap, which hinders it from being directly used by the host cell's machinery. So that's the difference of the plus and the minus sense. Again, difference between the plus RNA virus versus negative RNA virus. The plus RNA virus, you can just directly use their gRNA into their RNA as the mRNA. Meanwhile, the negative ones you need to make them into the complementary mRNA before you can use them before you can use them as a template to make the proteins. And so that's why RNA polymerase is packed in negative RNA viruses, okay? That's why they need their own RNA polymerase. Dun 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 dun. Let me just get the sticky note. Sticky note. Next, measles. 6 structural proteins. VSV encodes a glycoprotein that mediates attachment to the host cells, and etc. Measles in enveloped. Challenges for okay, let's mention this one. Transcription is controlled by stop-start signals, aka you have one promoter at the 3' end, and etc. What else? Gene order. Okay. So, this one's a good one. The genes that need to be transcribed in high amounts are usually nearer the 5' end compared to the 3' end. This one versus this, because the termination and reinitiation, they are not 100% efficient. So some polymerases will fall off. That's why the first few ones are replicated more or are made more. And that's what it means when the books say, where is it? Gene, position, affects mRNA, abundance. Yup, abundance. There you go. High qualities. Oh, and cytoplasmic infectious cycle of VSV. They replicate entirely in the cytoplasm and does not enter the host nucleus. And upon entry, the viral RNA genome is released, and transcription immediately begins. So again, VSV replicates in the cytoplasm and can't enter the nucleus. So, and then upon entry, the viral RNA genome is released and transcription immediately starts. Some concepts. No. VSV finished. Oh, single strand, negative sense, fixed order. Transcription proceeds from a single promoter. Gene order and start-stop signals make it difficult to insert new genes without disrupting the balance. And then, we have the steps in making a recombinant VSV. That's finished. Intergenic signals. Okay. Basic strategy for making recombinant. You isolate the VSV genome and then you create the cDNA clone. Then you rescue it. This one's finished. RNA synthesis reflects gene position. That's also done. Protein expression would mirror mRNA levels. Yes, because if you have more mRNA, then it's easier to make more protein. And then replication kinetics reveal growth defects in rearranged viruses. Because if the kinetics are bad, then that means they didn't grow well. Attenuated virulence in rearranged. Okay, then that just means if you rearrange the VSV, then its virulence would lessen. It's like you alleviated. It's synonymous to that. Oh, and there's also, if you rearrange the VSV gene order, it depends, but you can also reduce lethality. And for vaccines, the mice vaccinated with virus variants at varying doses, that's a lot of V, at varying doses would show different levels of survival after the challenge with high doses. So it really depends on the control level. So just as a review, how do adenovirus and VSP initiate and control gene expression after entering a cell? The adenovirus, let me write it as one more mini review. Adenovirus can use the host cell's machinery. Meanwhile, VSV would need its own RNA polymerase. So again, adenovirus, it is a DNA virus. It uses the host's RNA pol 2 to initiate transcription once it's inside the nucleus, and can just use the host. In contrast, VSV, its negative strand carries its own RNA polymerase inside the virion to just immediately transcribe its genome into mRNA. How do you study viruses in the lab? You can use cell culture and plaque assays. What is a viral vector? A viral vector is a virus that was engineered to deliver foreign material into cells. In viral vectors, you have foreign genetic material into the cells, and the vector is typically modified to remove its own replication or virulence genes, and then you insert your own gene of interest. What is genetic complementation and how is it used for working with recombinant viruses? So genetic complementation would be when your virus lacks a critical gene and then another virus rescues it and supplies the missing function. So the defective virus can then replicate and express its own gene. That's a really good thing to remember. Okay? So again, what is genetic complementation? Genetic complementation would be when, I have to write that. Genetic complementation. It occurs when a virus lacks a critical gene, so it needs a virus number 2 to rescue it, and virus 2 would supply the missing function. Alright. Next, how does the viral genome type determine the transcription pathway? That's finished. How are the different phases of adenovirus transcription controlled? It's temporally regulated. How does an RNA virus make messenger RNA? It uses its own RNA pol 2. What can we do with viral vectors? We can use them to deliver therapeutic genes or express CRISPR components for gene editing. And this one's finished. How can we experimentally distinguish early versus late adenovirus gene expression? So, early versus late expression. You can infect cells with adenovirus and then treat some samples with DNA replication inhibitor. So, early genes are expressed without DNA replications, but late genes need that. So, if you just harvest them at different time points, harvest the RNA or protein, and then use RT-PCR or Western, you can compare the expression levels. So early genes will still be present under the inhibition, but late will not be. So again, how if I want to see if early to distinguish early versus late gene expression, you can use an adenovirus. You have cells infected with an adenovirus and treated with inhibitors. And then what's gonna happen is early genes will still duplicate, meanwhile late genes will not. So at some point the early genes will be able to produce mRNA and proteins, and you can harvest the RNA or protein like extraction and then use RT-PCR or Western blot to check that. How can we detect the cytopathic effect? Well, you can just use cells, infect them with the virus, monitor them daily with using like a light microscope, and then if you want to quantify it, a plaque assay will work because infected cells will form plaques while in an otherwise intact monolayer. Okay, how can we verify that a recombinant adenovirus was successfully produced after inserting a transgene? Well, after you insert the modified adenovirus into 293 cells, you can monitor for cytopathic effects, which indicate viral replication. So you can isolate one more. Infect the cells and check the cytopathic effect. And then isolate the viral particles and do PCR to confirm the presence. And you can just check the fresh cells also by Western or reporter assays. How can we rescue a recombinant VSV encoding it? Just put in the gene of interest. Why is it necessary to package polymerase inside negative RNA viruses? Because the host has no enzyme to transcribe them. Period. And if you make a recombinant VSV that expresses a foreign antigen too close to the 5' end, could it be a problem? Yes. Foreign genes placed to the super near there could be. Too close to the 3' promoter, aka to the 5' end of the genome. They're transcribed at a very high level. So if you overexpress a foreign protein, it might slow the viral replication. So that's the mini summary for viruses. But yes, I think that is all for today. Thank you for watching. Bye.